Hello, thank you, Amy, and again, welcome everyone. To start off our talks today, our first speaker is Andrew Hammond, who is a visiting scholar at the Johns Hopkins Bloomberg School of Public Health and is based at Imperial College in London.
Thank you very much. So thank you to Jessica, Amy, Diego, Genevieve, Cecilia, and everyone who's made today happen. I'm going to jump straight in. I'm going to be talking today about some gene drive research that I've been doing, which took place mostly when I was at Imperial College London, and now I'm based at Johns Hopkins working on olfaction. So if anybody's interested in either of those two things, reach out to me afterwards. Should we go to the next slide? See, I'm not saying anything yet. Is this just me? Okay, there we go. I think you'll need to click to get it going. So I have a nice animation here. Okay, so there are over 3,500 mosquito species worldwide, but actually very few of these transmit disease to humans. And there are just 4 species that transmit the vast majority of malaria in Africa. And if you click, you'll see that these 4 species are actually Anopheles gambiae, Anopheles gallutii, Anopheles arabiensis, and Anopheles funestus. But now we have new tools, and we have new tools that allow us to maybe do something a little bit more interesting in terms of opening up new avenues for malaria elimination. And one of these new tools is genetic modification. So for this slide and the remainder of my slides, I have a theme where a blue mosquito means it's a normal unmodified mosquito. And if we click, we'll see that I have a red mosquito, and this means that it's been genetically modified. So if we click, what we'll see is that there are 2 ways that you could feasibly use genetic modifications to try and control malaria. And one obvious strategy is to immunize your mosquito. You could put in some sort of genetic vaccine that makes your mosquito unable to transmit disease. Or the alternative, if we click, is that you could introduce a modification that programs your mosquitoes to self-destruct. And this sounds very strange because we're going to try and introduce a genetic trait that will somehow kill them. So let's go to the next slide. So the problem with this approach is that if you were to make a genetic modification, something that you think is going to be useful and you think is somehow going to try and solve malaria, we have the problem that that genetic modification is only going to be in the mosquitoes you created it in. How would you get it into those mosquitoes in the wild? So with this theme, you see you have these 2 mosquitoes. One is modified in red and one's unmodified in blue. And if that modification was heterozygous, it will be transmitted to half of the offspring. So our red and our blue mosquito mate together, half of the offspring will inherit it and half will not. And if we go to the next animation, what you'll see is that over time, your genetic modification is not going to go anywhere. It's not going to spread. So yes, some mosquitoes will mate and they'll have some offspring, and that genetic modification would remain in the population. But over time, you're not going to see that population changing in any significant way. And if you wanted to do this on the scale that we see malaria transmission across sub-Saharan Africa, you would need to release hundreds of millions for the rest of time to have any meaningful effect. So if we go to the next animation, I want to introduce something called gene drive. And what gene drive can do is it can distort the normal rules of inheritance so that instead of being inherited at 50% frequency, your modification is almost inherited or maybe just biased inheritance. So the next animation, you'll see that every generation that your mosquitoes mate, your gene drive will, in theory, it could double. And it keeps increasing and increasing and increasing. And over time, you go from a very, very low proportion of your modified mosquitoes to potentially the entire population being modified. And the way that you count how quickly it spreads is through generations, not through real time. So we actually think about this in terms of mosquito generations, but it would also work if you were to attempt this strategy in a mouse or in anything else. It will take 20 to 30 generations to go from a handful to an entire population. And if we go to the next animation, you can imagine many different useful ways you could use this technology. You could use it to spread a genetic trait that makes your mosquitoes unable to bite humans, or perhaps unable to transmit disease, or the thing that I've been working on, which is something called a population suppressor to reduce the number of malaria-capable mosquitoes without affecting those species that do not transmit disease. So if we go to the next slide, the whole idea of a population suppressor is that you would be able to release your gene-drive mosquitoes just once or a limited number of times. You don't need to do a mass release. And that small release the gene drive will push that through the population for you without you needing to do the effort. And as the gene drive spreads, it will also bring down the total number of mosquitoes. And if we go to the next animation, the point I want to get at is that you do not need to eliminate the mosquito to eliminate malaria. If you can reduce the number of mosquitoes to a very low level, you'll find that malaria can actually be eliminated completely. So we go to the next animation, and I told you before that it takes 20 to 30 generations for a gene drive to spread, but a mosquito reproduces every 3 weeks. So 20 to 30 generations is actually very fast. Potentially that's just 2 years' time, even if you're released at less than 1% of the initial number of mosquitoes. In reality, we think it might take longer. It might take 4 or 5 years. And there are many things that make this more complicated. But in a very, very simple theoretical point of view, it's very, very fast. And we would expect it to be fast in a real release as well, not quite 2 years. So let's go to the next slide. So how can you spread something which makes your mosquitoes infertile? And this seems really very much like a paradox. So in the next animation, I'll show you 2 rules. The first rule is that you need to make your mosquitoes infertile if they're female. That's the first rule. So this gene drive is going to make female mosquitoes infertile. And the second rule, the next animation, is that it will only make them infertile if they inherit it from both parents. So female mosquitoes will be infertile, but not males. And it only affects those females if they inherited that modification from both parents. So let's look at the next animation. Let's see what happens. Our 2 mosquitoes mate together, and because it's a gene drive, it starts spreading through the population. Now that's great. And at this point, all of the mosquitoes would be fertile because one of their parents was always a wild one. One of them was always blue. But eventually they're going to start mating to each other because it starts becoming the dominant genotype in that population. So in the next animation, you'll see that when 2 of these gene-drive mosquitoes mate with each other, they'll produce both male and female offspring, but the females now, they're going to be infertile. So you might think, well, how does it spread? Well, the remaining mosquitoes, the next animation, the males and the heterozygous males and females will continue spreading that gene drive. And they'll continue spreading it faster than the suppression, which is killing those mosquitoes. It's always, the gene drive is always running faster than the destruction that follows it. So I think it's a very interesting idea and has great potential. So let's go to the next slide. So Austin Burt developed this idea in 2003, a very specific idea for gene drive using something called the driving endonuclease gene. It's been known as many different names, but I think nowadays everybody thinks of CRISPR gene drives and they think of what was initially pushed forwards by Professor Austin Burt at Imperial College London. So let's look at the next slide. How does it work? So you have these 2 chromosomes. And on the next animation, you'll see that this chromosome on the right, we've engineered it to contain this red gene. And the red gene is the gene drive. And it's simply 2 things. It's Cas9 and it's guide RNA. And I'm sure that many of you are aware of CRISPR right now, and especially with the Nobel Prize that was just given out to Emmanuel Charpentier and Jennifer Doudna. This technology has incredible potential to save lives. But in this case, we can engineer the chromosome to produce its own Cas9 and guide RNA. So on the next animation, what you'll see is we're going to program it to recognize the homologous chromosome, the partner chromosome. And so what's going to happen? Let's click the animation and see. This chromosome that we've engineered will produce Cas9 and guide RNA. And that will come along and it will cut the homologous chromosome and it will cut in this yellow sequence. So this chromosome is broken and it needs to be repaired. And the most efficient way to repair a broken chromosome is actually just to look at your partner and say, probably whatever my partner has is what I'm missing. So let's look at the next animation. That's exactly what happens. Okay, you have this copying over from one chromosome to the other, and the broken chromosome thinks that it's repairing itself perfectly, but actually it's just copied our modification inside. So that means that a mosquito that was originally heterozygous, it had one of your genes, it's now homozygous. So it's going to give your modification to potentially all of its progeny. And once this gene has moved inside, that's it. It's a irreversible reaction. So let's see the next slide, please. Okay, so I think you might need to click through to the next animation already. So the question is, do gene drives work? So there's one very quick way to test it. You can take a mosquito which has your gene drive and you can mate it to a mosquito that does not. And if you click on the next animation, what I'll show you is that in theory, you should have only 50% inheritance. So that would be this red line in the middle. That would be if there's no gene drive. But if the gene drive is working, you should get a boost, something extra. So let's see, when we tested it, how well did it work? So on the next animation, you'll see that we were getting 95 to 99% transmission of our modification. So this is far more than the 50% that Mendel would have predicted, and that's because the gene drive is working. So on the next animation, we performed some experiments and we said, okay, fine, we know that gene drives are working, at least in this very simple idea that they can bias their inheritance. But can they spread? What if we were to throw them into a population and just watch them? So we created a population of wild mosquitoes, and we threw into that population 25% of our gene drive males. And then we left them. We didn't add any more of our gene drive mosquitoes. We just let that population be. So let's see what happens in the next animation. So we release, and these two different populations in red and in blue, they started off at 25%, and we kept looking how many of them are there. And it went very, very quickly from 25% to 100%. It took only 8 to 11 generations. This is incredibly fast. And it's the first time that anyone had ever shown that a gene drive can modify an entire natural population. So let's go to the next slide. So what does it do? So I think we can click through a couple of these animations quite quickly, but I showed you before that we can cut this gene here, and this gene has a role. So let's click on the next animation. The role of this gene is that it is called the double sex gene, and specifically it's one part of the double sex gene called exon 5. And if we click on the next animation, what I want to show you is that this exon 5, this one little piece of DNA that we cut, it is essential for females to develop. So on the next animation, you'll see that a normal male and a normal female, they look different from each other. A male has these bushy antennae, and a female has this sharp proboscis. So on the next animation, you'll see that when we destroy this exon 5, you create these things called pseudomales. And they're not male and they're not female, and they're actually unable to bite, to transmit malaria or reproduce. So let's go to the next slide, please. So the gene drive spread. It reached 100% frequency in the population. And once that happened, what's going to happen? They're all going to be converted into these pseudomales. So let's look at the next animation. If they're all pseudomales, how can they reproduce? So if we can go to the next animation. Exactly. So we counted the number of eggs every generation, and it started off with 30,000. But within 8 to 12 generations, every female was converted into a pseudomale. The pseudomales can't reproduce, and the population crashed completely. So not only did this gene drive spread, but it completely eliminated those populations just by us putting in mosquitoes once. So if we go to the next slide. Yeah, I think this is the last real slide that I have. So we actually performed this experiment at a large scale. And we went into a semi-field testing facility, which is contained in Italy. And if you click on the next animation, the whole point of this was that we wanted to look at whether a gene drive can spread in harsh conditions, where there are overlapping generations. The mosquitoes need to be able to swarm. They need to be able to compete for mates. They need to actively search for food. This is really testing it at a scale that we say, once you get to this level, you consider that this may really be effective in a real setting in Africa. And we performed 4 different trials, and in each case, the gene drive spread from a very low initial frequency up to 100%. And it took about a year. So this was very exciting. And in all 4 cases, those populations were eliminated. So we're preparing this for publication now. We hope to submit probably within the next month or so. So if you go to the next slide, I think you can probably click through some of these and I'll just talk through them. But I think that hopefully I've convinced you that gene drives could be an important and potentially powerful strategy to control malaria. We see them as being complementary, not an either or. A gene drive should go with traditional vector control and any new technologies to control malaria. They're very fast, they're self-sustaining, they're very cheap, and that they will only affect those species that we target them to affect. They wouldn't spread to the other mosquitoes that do not transmit malaria. So I think we can go to my acknowledgements and just say there's an awful lot of people working on this project. It's more than 180 members. We have facilities that are being built and scientists being trained in Burkina Faso, Ghana, Mali, Uganda. Much of our team is made up of African researchers who are actually driving this technology forwards. And so this is very much a global effort to control malaria, not something that was just developed at Imperial College London. I think we can maybe leave the last slide for questions.
Wonderful. Thank you so much, Andrew. This is really exciting work. So there's just a few questions, and we'll have a few minutes for questions, but one simple one, hopefully, is at what point in development is the Cas9 expressed?
Yeah, that's very easy. We need to express that in the germline, so very, very early on, and it needs to affect the sperm and the eggs, but nothing else.
Okay, great. And then so more on like the ecological side of this and the impact it might have on it. For if a mosquito population goes down significantly during the dry season, would it be beneficial or distractive if a new population of mosquitoes moved in during like the next wet season? Thinking about areas that have high seasonality in their mosquito populations.
So that's a great question. Actually, what you'd expect is that the gene drive spreads very well when there are many mosquitoes, and when there are very few, it might fail to get in and those populations might recover. But anytime that they really recover and they become many, the gene drive is able to re-invade. So what you see is long time in Africa, you wouldn't have an elimination of the mosquito. It would never happen. What you'd have is you'd have anytime populations start to get large, they get suppressed. And when they become small and fragmented, they can become protected from being invaded by the gene drive. And so they'll keep coming up and down, but they'll never reach such high levels, high numbers of mosquitoes that they used to be.
And what do you think? There's a question in here about geographic spread of this. So if it's released in one area, how far do you, or could you anticipate this spreading to populations in other geographic areas?
So I think that from a regulatory point of view, you could not say that this could be contained easily to one geographical region, not the way that we've made this gene drive. There are other gene drive strategies where you could do that. And I think that's why when you look at this, you need to think about not just seeking informed consent from those people living in the area where you seek to do the initial release, but you need to get a broader consensus across Africa. And I think this is going to be an important part, a very important challenge in the development of gene drive technologies.